Welcome to the problem-solving set on aneuploidy. Problem 1. Use the word bank below to match each statement with the correct element or elements. Some statements may have more than one answer. Part A. An organism with 4 copies of a particular chromosome. So this is what's known as tetrasomy, so the individual would be tetrasomic. Now, if the individual was a diploid organism, that means that there's normally 2 copies of a particular chromosome. However, if there's 4 chromosomes, that means that its genotype would be written as 2n, which is the normal diploid, plus 2, where it's 2 extra chromosomes to give you a total of 4 copies of that one chromosome. B. Having one complete set or multiple complete sets of chromosomes. This is what's known as euploid. So, one complete set can be a haploid. Then humans have 2 complete sets of chromosomes, so we're called diploids. And then there's many other names for organisms that have more than 2 complete sets. Statement C. A condition of having an additional copy of a chromosome. So this is what's known as trisomy. And again, if the individual is normally a diploid, that means they have 2 copies of a chromosome. So if they have an additional 1 copy of that one of the chromosomes, they would be 2n plus 1. So this was actually in the word bank. Part D. The condition where both copies of a chromosome are missing. This is what's known as nullisomy. So in humans, we have 23 pairs of chromosomes, and nullisomy would be where we have, let's say, no copies of chromosome 4. So instead of having 46 chromosomes, we would only have 44. Okay, so that would be written as 2n minus 2 for a diploid. And you're minusing 2 because you're losing the complete set, which is a pair of 2, from the karyotype completely. Okay? Statement E. When one or a few chromosomes are present in too many or too few copies, and this is the general term for this. So this is what's known as aneuploidy, which is obviously the topic of interest for this chapter. So aneuploidy is where you have a complete karyotype, but then you maybe have an additional chromosome, an additional 2 chromosomes, or you're missing 1 or a few chromosomes from the complete karyotype. Now, for the next chapter, that's where we'll be talking about euploidy, which, as we saw in Statement B, is where you can have a variation on the complete set of chromosome. But this is just where you're having a variation to the set that you already have. Okay, and then finally, F, a condition where one member of a homologous pair is missing. This is known as monosomy. And for a diploid organism, this would be written as 2n minus 1, because you normally have 2 chromosomes in a homologous pair, but when 1 is missing, you're missing obviously 1, so it would be 2n normal diploid number minus 1. Problem 2. Chromosome aberrations involving the sex chromosomes can be described using aneuploidy terms. For women with the following conditions, determine which term can describe their chromosome content. So the first is a triple X syndrome, where a female has 3 X chromosomes instead of the normal 2. So this is what's known as trisomy. And again, this is where you have a diploid organism, a female. Normally she would have a pair of X chromosomes, so just 2, but she has an extra one, which is where the tri for 3 comes in. So her chromosome content can be written as 2n plus 1, the normal chromosome for a diploid plus an extra one, and this is where the X chromosome has 3 copies. For Turner syndrome, this is where a female has only 1 X chromosome, and it says XO, but that's really a 0, meaning there's no chromosome there. So this is what's known as monosomy. And this chromosome content can be written as 2n, which is the normal diploid content, minus 1, because you're missing one of the chromosomes from a homologous pair of XX. So X has only 1 copy. And I'll write for both of these obviously something you know, but we might as well write it down that normally there's 2 X's for female. Question 3. Color blindness is a human X-linked recessive condition. A young male with Klinefelter syndrome, karyotype XXY, and his brother, karyotype XY, are both colorblind, but their parents have normal vision. Describe the non-disjunction event that occurred to give rise to the brother with Klinefelters. So we know that the Klinefelter has XXY karyotype, and because color blindness is a recessive condition, both of his X chromosomes must be mutated with this colorblind allele. So his karyotype is actually, let's write it as XC, XCY. And we want to know what non-disjunction event can give rise to his karyotype. So we know that the mother and the father both have normal vision. So for the father to have normal vision, he must have a completely normal X chromosome and a Y chromosome. Now the mother, although she has normal vision, she must be a carrier of the colorblind X chromosome, or else else would her son get, both sons get this colorblindness mutation? So we know that her karyotype must be X, normal X, XC, so she is a carrier. And therefore, because we know that the father doesn't carry this colorblind allele, we know that the non-disjunction event must have occurred in the mother. So non-disjunction definitely occurred in the mother. But now we have to figure out at what point in meiosis did this occur. So let's draw just an overview of meiosis in terms of these X chromosomes from the mother. So at meiosis 1, in anaphase, when they're separating, you have this tetrad, which is normal X, the 2 sister chromatids, meeting up with XC, XC, the other pair of sister chromatids. Now, this would go through meiosis and would yield one cell having the 2 normal X's and another cell having the 2 colorblind X's. Now at meiosis 2, when these separate, this would yield to gametes having normal X, and then 2 colorblinds. Now this is for normal meiosis. Now let's look at meiosis 1 and 2 and see what the non-disjunction would look like. Okay, so for meiosis 1, if this tetrad didn't come apart, that means that you would have a cell with still the 4 chromosomes in it. However, when they lined up in meiosis 2 to separate, the 2 sister chromatids would line up separately, so they wouldn't line up as a tetrad anymore, and therefore, when they go through meiosis 2, it would yield gametes that have a normal X chromosome and a colorblind X chromosome, because remember, they aren't considered tetrads anymore, they're considered just pairs of different chromosomes, so they separate out differently. So that's what would happen from non-disjunction 1. Now from non-disjunction 2, so you have these 2 cells here, so at meiosis 2 you have a cell with XX normal and a cell with the 2 colorblind X's. And if you have non-disjunction here, which would be called non-disjunction 2 for meiosis 2, you would get gametes, one that has 2 normal X's and one that has no X, one that has 2 colorblind X's, and another that has no X. So obviously here, this is the gamete that fused with a gamete with a Y chromosome from the father to make this Klinefelter son who was colorblind. So therefore you know that the non-disjunction event that occurred was non-disjunction 2 for meiosis 2, and it occurred in the mother. Problem 4. In a plant species, the dominant allele P+, which we can just write as plus, produces red flowers, and its recessive allele P produces pink flowers. Plants trisomic for this color-determining chromosome can produce N and N+1 functional female gametes or seeds, but the male gametes, pollen, are only viable as N. Determine the expected phenotypic ratio for each of the following crosses. So, let's look at part A. The seed parent, as they tell you, can make viable gametes that are either N, which has only one chromosome, or N+1, which has two chromosomes for this color-determining chromosome. So let's look at the seed parent and what gametes they can make first. So obviously they can make gametes that have only N or one of these three chromosomes. So it could be a plus gamete, another plus gamete, and a P gamete. And the reason why I'm drawing plus twice, even though they give the same phenotype, they are distinctly different chromosomes, so we're going to have to include them in the probability. Okay? But then they can also make viable gametes that are N+1, so you can have this first plus, you can have plus P, which has the first plus chromosome, and you can have the other plus P. Okay, so a total of 6 gametes that this seed parent can make. Now you're crossing it with a pollen parent, and remember that pollen is only viable as N. So the pollen gametes can either be plus, or P. And now for the cross, let's look at the offspring, the F1 generation. And we're just going to cross each gamete. So we'll cross this first plus gamete with these 3, and then we'll do the same for the others. So the offspring, remember that each of these is a 1/6 chance of happening, and this is a 1/3 chance of happening. So you get a 1 out of 18 chance of getting this first plus, 1/18th chance of getting another plus, 1/18 chance of getting plus P. Then this plus is a 1/6 of happening, so again you're going to get these 3 offspring again, but because they're including different chromosomes with the same phenotype, remember, but there are different chromosomes, you have to write them again. So 1/18th for each. Okay, now you have P, so 1/18th P plus, 1/18 P plus. Okay, so these accounted for these 3 N gametes from the seed parent. Now let's look at the N+1 gametes. So again, all the probabilities are going to be 1/18th. So you have plus. Okay, 1/18th, so that took care of that gamete. Now you have plus P plus, 1/18 plus P plus, 1/18 plus P, and that took care of that gamete. Then you have 1/18 plus P plus, 1/18 plus P plus, and finally 1/18 plus P. Okay, so these are the genotypes. What they want, however, is the phenotypic ratios, right? So let's look at what these alleles confer. So the dominant allele is the plus, and that produces red flowers. And the recessive allele is P, and that produces pink flowers. So any of the offspring that have at least one plus wild-type dominant allele will be red. So let's see, any of the ones with a plus will all be red. So I could draw that here, so red. This is the first one that's going to be pink. And here, red. Red. Okay, so as you can see, if you count them up, you have 17 red to 1 pink. So this is the only pink. So the total for part A would be 17 red to 1 pink. Okay? Now let's look at cross B. So cross B, the seed gametes, remember, can be n or n plus 1. So it can be plus P, P, plus this first P, plus the second P, and P, P. So all of these have a 1/6 chance of occurring. And then the pollen is only viable as n, so this will be plus, plus, or P. So again, you're going to do this cross. So each of the seed gametes are going to be crossed with each of the pollen gametes. And again, the pollen have a 1/3 chance of happening. So again, your offspring going to be 1/18th chance of each formation. Okay, so let's just go through this quick. Plus, 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 1/18 plus P. That takes care of that gamete. Now, because these two are both P, I'm just going to shorten up a bit. So, as you know, they'll all give P plus, P plus, PP. So instead of doing 1/18th for each, let's do 2/18th for each, because we know the bottom one is going to be the same. So we have 2/18th P plus, 2/18 P plus, 2/18 P, P. That takes care of both of those gametes at the same time. And here, these two gametes are also the same, so let's do the same thing. So we'll have 2/18th plus P plus, 2/18 plus P plus, 2/18 plus P, P. That takes care of both of those, and then you have this final gamete. So 1/18th now, P, P plus, 1/18 P, P plus, 1/18 P, P, P. So again, let's run through and look at for the phenotypes. Any offspring with a plus chromosome are going to be red. Okay, this one will be pink. I'll circle them. Red. And this one's also going to be pink. So now when you count these up, you have 1, 2, 3. Remember to count this twice. So 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So you have here 15 red to 3 pink. So you can simplify that down if you divide both by 3 to 5 red to 1 pink. So this is the phenotypic ratio for part B. So you have 5 red to 1 pink. And I'll write that on the front page so you have everything together. So okay, now let's do cross C. The seed parents, the gamete can be plus, plus, P, and then plus, plus, plus P, plus P. And the pollen can either be plus or P. Now here the pollen is just normal, it's not trisomic, so that changes it a little bit. Now the probability is 1/2 for each of these gametes, but these are still 1/6. So now instead of 1 out of 18th for the offspring, it's going to be 1 out of 12. Okay, so let's do this cross. So again, these two first gametes are the same, so I'm going to use the same simplification as before. So instead of it being 1/12 will be plus plus, it's going to be 2/12. And then it would also be 2/12 plus P. And that takes care of these two gametes. Then you have P, so it'll be 1/12 P plus 1/12 P, P. Then you have plus plus, so 1 out of 12 chance plus 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 1 out of 12 plus plus P. And then these last two gametes are the same, so let's simplify that again. And it'll be 2/12 plus P plus 2 out of 12 plus P, P. And the phenotype, anything with a plus will be red. So this is the only pink. So if you count up, you see 4, 5, 6, 7, 8, 9, 10, 11. So you have 11 red to 1 pink. And then finally, let's do the last cross. So here the seed gametes can be plus P, P, or plus P, plus P, P, P. All have a 1/6 chance of happening. And the pollen, again, here is just normal. It's not trisomic, so it can either be plus or P, which each have a 1/2 chance. So let's do this. So 1 out of 12 plus plus 1 out of 12 plus P. The next two gametes are the same, so let's do our simplification trick. So it'll be 2/12 P plus 2/12 P, P. That takes care of both of those. Then you have two that are the same again. 2/12 plus P plus 2/12 plus P, P. And then finally 1/12 P, P plus and 1/12 P, P, P. Anything with the plus is going to be red phenotypically. So here you're going to have 3 pink. So you have 2, 3, 4, 5, 6, 7, 8, 9, 9 red to 3 pink, which you can simplify down, divided by 3, to 3 red, 1 pink. Okay, so you have these answers to the right for each of the crosses, and you can see when aneuploidy comes into play, you get a wide variety of offspring phenotypic ratios.